Hello, my name is Sahil Bave, and welcome to my genetics final project. To understand this genetics final project, you would need a basic understanding of what sourdough starters are. Well, sourdough starters are a combination of all sorts of things. They're a combination of water, flour, wild yeast, and air. All of these are contained together in a closed container. Long ago, these four nations lived in harmony until the wild yeast attacked. This is a Avatar reference in case you're wondering, but that's besides the point. The wild yeast attacks in the sense that it uses the water, flour, and air all to grow as it metabolizes the materials found inside of it to produce more cells and what we call cellular growth. The reason I'm mentioning growth a lot is because that is the overall goal of sourdough starters. It is to grow yeast and to let the yeast metabolize the materials found in the sourdough starters. Here are pictures of sourdough starters. The starter on the left is a newer, runnier sample, as it has not had the time to let the yeast grow and metabolize, while the picture on the right showcases a older sample where the yeast has grown and metabolized a lot of the materials that were originally found in the sourdough starter. Now, how does this relate to genetics? Well, sourdough is actually a perfect example of genetics, as the materials that are used to cultivate and metabolize these sourdough starters are prime examples of genetics. These are bacteria and yeast that all work together to change the original sourdough starter that we saw on the left in the previous slide into the sourdough starter that we saw on the right. And we use KBase and other bioinformatics tools to analyze the bacteria and other organisms found in these starters. But to understand this, you need to understand bioinformatics a little bit. Bioinformatics is the science of collecting and analyzing complex biological data, such as genetic codes. So, we are taking the genetic codes found in the sourdough starters and analyzing them. That is what KBase is helping us do. Another thing we use KBase in bioinformatics for is DNA sequencing. DNA sequencing is a process of determining nucleic acid sequences, or the order of nucleotides in DNA. As you know, there are four types of nucleotides in DNA: thymine, adenine, cytosine, and guanine. The order of these in certain molecules, or not certain molecules, in certain organisms is very specific. And once we find the order, we know the genetic code of the organism. And this allows us to determine what specific parts of this genetic code can be considered genes. And once we find the individual nucleotide base pairs that make up these genes, we can manipulate them in a way that allows us to find the function and the purpose of these genes. Now, let me show you my results. My genetics lab was online, so I did not make sourdough starters, but I did use KBase and other bioinformatic tools to analyze the starters. What was present? Well, there were many different bacteria present. We had two groups, the salt group and the control group. The salt group, obviously being the saltier group, had more gram-positive and bacteria and organisms that can survive saltier conditions, whereas the control group had more gram-negative bacteria that were less able to survive salt conditions. The control group is what is normally expected of sourdough starters, and the salt group was used in contrast. Here we see the different species found in each of these starters. On the left, you see two columns. The leftmost column is the salt column, containing the organisms found in the salt DNA, or the salt sample, and the right column is the control column, which is the organisms found in the control group. And on the right of these two columns is the key with lots of different organism names. These organisms are the organisms that make up both the salt and the control sample. And they are labeled in order of most available or most concentrated to least available or least concentrated. This means that the higher up the list a bacteria or an organism or a virus is, the more likely you are to find it in either of these samples. In each of these columns, they show blocks indicating how much percentage of the reads were considered in each of these colored bacteria. For example, the salt sample shows a great affinity for Chlamydia abortus, meaning a large portion of the salt sample was Chlamydia abortus due to the percent of the reads. Now, what is the next step? Well, it would be to change the salt concentration. The amount of salt, of course, affects the environment in which the organisms can grow. So changing the amount of salt would change the types of organisms that can grow. So we would want to set up an experiment based on salt concentration and keep a control group. So what does that mean? That means that we must have one control group, an unsalted group, as before, but instead of having just one group that has salt, we'll have multiple groups with salt. A group with a small amount of salt, a group with an intermediate amount of salt, and a group with a large amount of salt. This will show a contrast between having too much salt, having too little salt, and having not any salt at all. Of course, this allows us to compare bacteria results, and we would have to do more bioinformatic sequencing and bioinformatic data analysis, but that is all expected in genetic labs, because the overall goal is to see how salt concentration affects a sourdough starter in small amounts, moderate amounts, and large amounts, as bacteria growth in sourdough starters is the most important aspect. This concludes my presentation. I hope you learned a great deal about sourdough starters and genetics. My name is Sahil Bave, and I am glad to have hosted you for this presentation.